All right, thank you again to our presenters for their wonderful presentations. We'll go ahead and start our question and answer panel to include all our presenters from earlier, and also joining the panel, we have Dr. Hansong Chen, a senior interdisciplinary scientist from the Division of Product Quality Assessment 12, within the Office of Product Quality Assessment 2 from the Office of Pharmaceutical Quality. So thank you again for joining us. As before, attendees in the room are invited to line up at the mics to ask your questions. And then for those online logged in, if you haven't had a chance to submit your question already using that on-screen link and QR code, please do so now. And we'll answer as many questions as we can. So first, we'll see if anyone in the room, again, wants you to come up to the mic. If not, in the meantime, we'll go to our first question online.
Our first question is for Dr. Yang Liu or Dr. Hansong Chen. How was IV-IVC for mesalamine able to capture variability? How many different rates of release formulation were assessed? And can you provide more information on unit impulse response formulation used for deconvolution?
Okay, so can you repeat the second part of the question, please?
Sure, yes. The second part was how many different rates of release formulation were assessed? And then can you provide more information on unit impulse response formulation used for deconvolution?
Yeah, I think maybe Fang is the expert for IVFC, so I'll let her just chime in first.
I think the first question is about the variabilities and also how many immediate delayed release formulations assessed. I think for this one, we focus on the mesalamine delayed release tablets. The ROD is Liada. So we focus on these ROD and the relevant generic drug formulations. We use these as a case example for indicating how PBPK can be used to identify the biopredictive dissolution and predict local drug amount of drug in the colon. So yeah, and regarding the unit pulse for the disposition part, I think they are asking about the disposition. Actually, we use the two-compartment model and to get the disposition on the PK parameters. You know, the two compartments, we have K1 to K21 and also the clearance that we put into the model and to estimate disposition parameters. I hope that answered the question.
Yeah, thank you, Fang. I'll also chime in a little bit more from the formulation perspective. So for this drug, mesalamine DR tablets, referencing the same ROD product, there have been several endo versions or generic version approved by the FDA. From a formulation perspective, these formulations include the one we have in our case study. They all use very similar excipient, the pH-dependent excipient per se. However, these approved end products for the pH-dependent excipient they use, they have different amount or they have different ratio. So if we're looking back into the in-house data we have been reviewing for this generic product, we are observing some very different dissolution profiles, either between the ROD and the test or across different endos. So that also reflects the variability of this kind of a complex mechanism of release. However, as Fang just mentioned, these data or this variability or the slight different design of the formulation were taken into consideration when we put the in vitro dissolution data into the PBPK modeling to get a supportive evidence as one of the piece of evidence we use to support either the approval or the decline of the end.
Thank you so much for all that information provided. We'll go to another online question.
All right, this question is directed towards Dr. Likan Liang. For products like defibrotide, which is a polydispersed mixture, and RLD is expected to have a lot of variability, how is sameness comparison performed if the RLD itself is changing?
Thank you for the extremely good question. Actually, I didn't have time to talk about defibrotide. Defibrotide is a little bit different from the other oligonucleotides because the RLD sequence itself is not defined because it's a mixture of different combinations of sequences. This is by control depolymerization of the DNA from the toxin. So, I mean, itself has a combination of all different sequences. So instead of trying to compare the sequences, which is very challenging, actually we published guidance and actually advising the industry to actually attack this from different angle. If you use the same source of the, the animal tissues or organs and things like that, from the same source, and also use a similar manufacturing process, the same mode of depolymerization, you may have a better chance to have similar kind of distribution on the sequences. So this is quite different from other oligonucleotides. Yeah, thank you. I would highly recommend that you check the PSG for advices. Thanks.
Thank you again for that response. A reminder again to people in the room here, remember, this is your opportunity to get your questions answered right here, right now. So feel free to come up again to the mics. If we don't have any takers for the moment, we'll continue with our online questions. Our next one.
All right, we have one for Dr. Yan Wang. Is it acceptable to establish and submit IV/IVC analyses using animal models for LAI products due to the long half-lives for LAIs?
Yeah, thank you for that question. I guess the key in that question is the animal data. So I think as of now, animal data, how we view for generic development, we are certainly open, to get that piece of evidence as like supportive, or applicant could use that. And that's what we have been doing in our own research to obtain a baseline understanding of what may be the drug release mechanism and behavior, although species translation is always technically challenging. But as of now, if it's intended for regulatory purposes, I don't think animal data is accepted to support generic assessment or approval. But for development purpose, we are open to look at the data, for instance, in pre-ANDA meeting mechanism and provide feedback to help maybe better design the PK or whatever is relevant to the development.
Thank you again for that response. We'll go to another online question.
Of course, this one is directed towards Dr. Hansong Chen. Is it acceptable to develop a dissolution or IVRT method different from the USP monograph ones?
Thank you for the question. Yeah, this is very general question. The short answer is yes. Yeah, you can develop a different method from the ones listed in the USP monograph. I would like to add a little bit more about this. Our current thinking is, the dissolution method or in vitro drug release method is product specific. So, when you develop the dissolution method, the USP method or FDA database method, it's just the starting point. So you should develop your own method that's appropriate for your drug product. So our expectation is, the method should be sufficiently discriminating against the CMS and the CPPs. So ideally, the method should be clinically relevant. Yeah, thank you.
right, thank you for that information. Again, still open for anyone in the room to come up if you'd like. But if not, in the meantime, we'll still go to another online question.
All right, we have one for Dr. Fang Wu. For mesalamine, if PBPK modeling shows that dissolution in media less than pH 7 is not as relevant, can the recommendations be changed to remove dissolution testing in these media?
Thank you for the question. So yeah, as we showed that for the LEADA and this specific drug product, the dissolution at pH less than 7 appears not be predictive. However, that this dissolution data submitted to the agency still can be considered as some like supporting evidence, or we can also consider look at the data and to see whether there's any difference, and also we consider the release mechanism about these drug product. And then yeah, for this product we use the biopredictive solution mainly to put into the PBPK and predict the local drug amount. So we still need that the data at less than 7 for looking at the data. That would I comment. Thank you.
If I may, I also want to provide a little bit comments for that question. So yeah, so as I like what Dr. Wang and also Dr. Wu was talking about, and the PSG actually is reflecting the current thinking or the growing understanding on certain aspects based on the knowledge and experience we collected in our review, in our internal research job. Regarding the mesalamine DR tablets, dissolution data under conditions that is a little bit more acidic. So my comment is, so the recommended evaluation stages in the PSG with the 4 different pH values is try to represent all the possible conditions after the drug actually was delivered into the small intestine and after. So this is, yeah, you can say this is a more conservative approach. If a test product can have a very similar dissolution in all these conditions, that would be the ideal case. However, just like Fang mentioned, if the applicant observes some issues or some incomparability between the test and the reference in, for example, 6.8 pH condition, it's still recommended to submit the data, and the agency in our review or in the follow-up meeting with the applicant, we are open, just like the case study we shared today, we are open to consider and take into account other evidence, including but not limited to PBPK modeling, to support and to evaluate the bioequivalence from a totality of evidence perspective. So hopefully that is clear. Thank you.
Thank you so much for that response. And it looks like we have a question in the room. Go ahead.
Yes, thank you, Marie Ducharme, Learning Confirm. It's a follow-up to the previous question that was online. It seemed to me that it made sense that you've proven that 6.8 was not predictive, that you would remove it from the individual product guidance. It seems to me that made sense. Going back to your answer, where you said that the 6.8 was not predictive, here is a question for you. In your PBPK analysis, if I understand correctly, you kind of said that the predictions were validated because you were plus or minus 20% on the mean. And I think I heard that you decided that it was not predictive because when you included the 6.8, you had plus or minus 30%, okay? My question is, this product is highly variable, right? Correct? It has 70% to 100% intra-individual variability, correct? So why would plus or minus 30% not be good when it's highly variable at plus or minus 70 to 100%?
Yeah, so first I would like to clarify, and just now we said that although we showed that dissolution at pH 6.8 is not biopredictive, but this is still included in the current PSG And also we are current thinking is that that data can still be submitted to the agency, and we are going to look at it for evidence and considering the study, the formulation design, release
Yeah, I
mechanism.
understood that.
Yeah,
I understood
so
that.
just
Yeah.
not to be removed from the PSG, just to clarify that. And then for the variability you mentioned, yes, the mesalamine, it is a highly variable drug product. And what we generally consider, which is biopredictive, is based on our current practice on using PK modeling, especially for the BE assessment. If the prediction error is more than 20 or 25%, that considered is not a good prediction. So that based on that, we considered that the pH 6.8 included that is not biopredictive as compared to the observed PK. Yeah, and yeah, I understand.
I'm sorry, but in previous presentation by the agency, validation of PBPK is usually plus or minus twofold.
So yeah, I think
so I'm really surprised that you're accepting plus or minus 20% and then rejecting plus or minus 30% when this is a product that's highly variable. I mean, it's a little bit surprising.
Yeah, it's not rejecting, just comparing to the other dissolution that they are.
But that's how you decided that 6.8% was not, that 6.8 was not relevant. That's how you decided that, because you got plus or minus 30%. I mean, think about it. I just, anyway, a comment for thinking.
Yeah, thank you. Thank you for the comment. Yeah, for the twofold, actually, we are considering whether it is validated or not, depending on the modeling purpose. The twofold, yeah, as you know that the DDI, when we use the modeling to see whether it is validated or not, sometimes twofold we can consider, and also if there is a the platform validation, we can consider that big variability, the prediction error. But for the PE assessment, generally we consider like 20%, around 20%. Yeah, thank you.
Thank you for that. We have another question here. Yep.
So I have a question for the PLG, the long-acting drug. So the PLGA. The PLGA is an excipient, and there's a lot of study, a lot of focus from a talk. So from a previous talks, especially yesterday, it's API has been focused a lot. Your talk, PLGA, has a lot of research study there. So my question is, what reason behind that? Just you want to make sure the release duration is okay, or you also talk about impurity or something else? Why that has so much focus?
So your question is why we spend so much efforts on characterizing the PLG, considering it's being
just
an
excipient.
excipient?
Yep.
Okay. Yeah, I think that's a fair question. So first of all, PLG is the excipient, and it's actually a functional excipient, meaning its characteristics plays a very important role for the drug product performance. Then the main reason for us to spend so much efforts on understanding the polymer, one part is because its impact on the product performance, then that directly relates to the successful rate of having a successful product. But the other hand, as I mentioned, is to fulfill our regulation. So the regulation, because the PLG-based products, most of them are parenteral products. So the regulation requires the generic to demonstrate the composition of the generic is qualitatively and quantitatively the same. But then for PLG polymer, because of the inherent variability, is not as straightforward as some excipients. Q1, it strictly goes to chemical identity. So our efforts is really to understand the characteristics of the polymer and the source impact of the polymer on the product performance, and also to guide. Right now, we have made great progress in terms of we have clear guidance to tell the applicant what they should look for, what they should do. But then having said that, we also understand from generic development standpoint, it's still very restrictive in terms of today, probably in terms of characteristics, the molecular weight, we have probably the max flexibility depending on the product and our understanding. So if we know a product, the PK and its release is not so sensitive to differences in molecular weight, mean molecular weight and distribution, then on face value, if we see a 10 to even 20 kD difference in the mean molecular weight, we would still consider that as the same polymer. Then, but in some other cases, depending on the product or dosage form, we also know if you have less than 5 kD difference, you will see a difference in the release and the PK. Those things may not be obvious to applicant depending on how much experience they really have. And that's why we do those research and publish, share our understanding, and also we provide as much detail recommendations in our responses to their inquiries to facilitate their development, to improve the successful rate, because those are so complex and costly to develop, they probably don't have many chances for failure.
Thank you. I guess we can talk a lot on that in offline.
Yeah, sure.
All right, thank you again for that response. Maybe we'll try to fit in one more online question.
Sure. So we have a question for Dr. Yang Wang. So are there any validated mathematical models for establishing IV-IVC for long-acting injectables?
A very good and challenging question. As of now, I think the IV-IVC we have successfully demonstrated in animal, those are more empirical mathematical model. So either time shifting or time factoring based on mostly, the release mechanism. If it's sigmoidal triphasic release, we would try to keep the shape of the in vitro, then just compare the time scale and factor that. Having said that, there are ongoing efforts actually in the sister division, DQMM, to look into PBPK, for instance, those model to establish IV-IVC mechanistic modeling. And also from our perspective, like I mentioned briefly, we are also looking to like the physics, the AI-based simulation, and then the imaging microstructure supported release mechanism exploration. And we are looking to using those tools to establish IV-IVC. But at the moment, I don't think we have a robust validated mathematical model that can fit all products.
All right, thank you again for that response. And I know we're going a bit into lunch break, so maybe we'll go ahead and end our session there. But thank you again to our presenters and panelists for all the excellent information and insights. Let's go ahead and give them another hand. Yeah.